the role of senescent cells in the infection. How did COVID go from a disease transmitted by eating wild animals to one transmitted through the air? You can answer that one, okay?
Yeah.
All right. Okay. There's a question about Nevin's study has identified a catalog of human proteins. Prohibiting these interactions could result in novel drugs. Do you favor any of the identified interactions? So you could talk about. Do you see promise in widespread? Do you see promise in widespread antibody testing?
All right, Chris, we're live in about 30 seconds.
Okie dokie.
Yeah, Chris, you don't seem to hear me. Can you hear me?
I can hear you.
Okay, yeah, yeah, those questions are fine.
Okay. Give me a signal, Mark. I got my script up.
All right, everyone, we're live.
All right, thank you. Hello, everyone, and welcome to our first webinar focused on the COVID-19 pandemic. I'm Chris Rebelo. I'm the Director of Communications at the Buck Institute for Research on Aging, and I'll be moderating today's conversation with Dr. Eric Verdin, our President and CEO. We do wish we were coming to you at a happier time, but we are glad to bring scientific expertise to this public health crisis. We have 5 eminently qualified experts lined up for this web series, and I hope you'll be able to join us for all of them. Excuse me. I also welcome you to be an active participant in these sessions. You should see a chat widget at the bottom right of your screen. Please use that feature if you'd like to pose a question for Dr. Verdin during today's session. Eric has been at the helm of the Buck for nearly 4 years, and he brings a unique combination of expertise to this crisis. Before coming to the Buck, he was a senior investigator at the Gladstone Institute for Virology and Immunology. And here at the Buck, his lab studies aging and the immune system, an inquiry that has taken on much more significance during the COVID-19 pandemic. So let's get into it. Hi, Eric.
Hi, Chris. Good morning.
Good morning. So I understand you have some folks on your team that crunch numbers. Where are we right now as far as this pandemic goes?
So we are at quite early, and I know that this is probably not what our audience wants to hear, but we're quite early, and three different models have emerged from looking at the world. Actually, one model is what happens if we let this virus roam free, and this is what we see happening in Spain, in Italy, and unfortunately in New York. That's the model of the consequence of what would happen if we act late. The second model is what has happened in some Asian countries. Singapore and South Korea are actually good examples where there's been very active and aggressive public health measures which have largely controlled the infection so far. And finally, the third model is the model of California, where we have acted early, but in a different manner, which with a lockdown, which obviously brings its own set of economic consequences. So I think right now it's clear what works. It's clear what are the consequences of each of these actions. And I think we're going to have to really decide which way we move into the future.
But it does look like California did the right thing early on, and we are in better shape than we expected, right?
Yes, the lockdown clearly works, especially when applied early. There's clear evidence that the numbers in California and especially in the Bay Area are really flat with very low level of mortality and what appears to be a low penetrance of the virus in the population.
All right. So let's talk about older adults, because it's obvious they are at higher risk for COVID-19. They've had more mortalities. Why are older adults more at risk for this disease?
Well, it's not only older adults. It appears to be, of course, your age is a direct predictor. The older you are, the more you are at risk, not so much for the infection, but for the bad complications and the fatalities associated with the infection. But again, it's not only in older people. It turns out that obesity is also a really high risk factor for complications. And so are a number of chronic conditions that we have known for a long time to be a number of chronic conditions associated with aging. And these are heart disease, heart attack, stroke, cancer survivors, type 2 diabetes, chronic obstructive pulmonary disease. So this whole group of conditions we know is associated with aging, but some people can actually get them earlier. It turns out that people who develop these complications early are also at increased risk for COVID-19.
So what happens to the immune system as we age?
So that's actually is really central to, we think, is central to why older individuals and those who have these chronic diseases become more affected. So the thinking is, and maybe I'll speak a little bit about immunology. There are two broad arms to the immune system. The first arm is the immediate response that our organism have against any invading pathogens, bacteria or viruses. It's called the innate immune response. It's the first line of defense. It is non-specific. That is, it's not targeted towards one virus or the other. It just protects us globally. But that protection obviously is not complete, and viruses and bacteria have evade actually this initial response. So that response, early and non-specific, is actually what becomes dysregulated during aging, hyperactive. Now, and this is a phenomenon that people have called chronic inflammation or inflammaging. And interestingly, this is actually a driving factor for these chronic diseases of aging. It's not only about aging itself, but heart attacks, type 2 diabetes are all driven in significant part by this chronic inflammation. So that response is hyperactivated. In terms of the second part of the response, the immune response, which is really important and will become particularly important when we develop a vaccine, is the adaptive immune response, which is highly specific and targeted against the virus. And so obviously right now, since no one is immune to this virus, we are all relying on this initial defense, which you might think is actually a good thing. If it's hyperactive, we should be more protected. It's a good thing initially, but it becomes a problem later in the infection, because we think this is what actually causes the death, is this overactive immune response.
So even though older adults, our immune systems might be a little tired out from time and stress, we're still able to mount this hyperactive immune response in some people?
Yes, and it seems that what differentiates the people who actually are doing well and not doing well after infection with this coronavirus are those that mount an appropriate immune response, that is most of us, and those who mount a disproportionate response. Actually, the response itself becomes the problem. There's a phenomenon that people have called the cytokine storm,
Yes.
which is the storm is made of these cytokines, and these cytokines are chemical mediators that the immune system uses to signal the emergency and to actually to induce a response. So that response is disproportionate in older individuals. And we think this is one of the problems.
So keeping on talking about the immune system, everybody, well, a lot of researchers around the world are focused on developing a vaccine for COVID-19. So I think everybody's assuming that we will have a vaccine at some point in time. But I'm wondering about older adults. I have read that vaccines often fail in older adults because our immune systems are weak. Do you think that, is that going to be an issue for any vaccine that might be developed?
That's a very good question. So I just told you about the innate immune response, the early non-specific response. Vaccines are dependent on this second part of the immune response, which is called adaptive, which is highly selective. And we know from previous experience with other viruses, in particular the flu, that the older you're getting, that adaptive immune response becomes deficient. And that could also be a significant problem in terms of vaccination. And we know, for example, in terms of the flu, depending on your age, and for people above 70 years old, only 20 to 30% of people respond in an appropriate manner. And so here comes the concept of herd immunity. The way vaccines work typically is not so much by protecting everybody against the virus, but is also limiting the spreading of the virus. And so the way the flu vaccine works in general is, especially for older individuals, is by restricting the circulation of the virus in the population. And so even though there is a concern that older individuals might not respond to a COVID vaccine, SARS-CoV-2 vaccine, they should be protected by herd immunity when we get there.
So that would mean that older adults probably will need to take precautions longer than other folks while everybody catches up to being immune from this disease.
Absolutely. So a lot has been said and a lot of hope is being placed in terms of what this vaccine is going to do for us. I think, we should be cautious in our optimism. The good news is that coronaviruses are immunogen. It's not like HIV, so it is very likely we will be able to develop a vaccine. The question is how soon, and the second question is how soon will we be able to deploy it on the scale in which we are looking for to protect everyone. And so until that day, it is, and even after that, it will be highly recommended to older individuals and those at risk to keep some degree of social distancing. I know this is not what anybody wants to hear, but this is the reality of what we're going to have to deal with in the coming year and a half.
Got it. So I want to go to a question that somebody posed online. So it talks about the publication that came out of UCSF being led by Dr. Nevin Krogan, who's going to be with us next week, that he identified a catalog of human proteins that interact with the virus that causes COVID-19. Prohibiting these interactions could result in novel drugs stalling the viral replication. The buck is involved in this research now, and the question is, do you favor any of the identified interactions? So can you talk about what's most promising and what you're working on?
Yeah, so this is an exciting development. So Nevin Krogan and Melanie Ott are both adjunct faculty here at the Buck, and both work at UCSF and at the Gladstone. Nevin, as you said, identified a large number of cellular proteins that the virus latches on. So think about a virus as a parasite. It comes into the cell and it needs to replicate itself to make new virus. And the way it does this, because these are relatively simple genetic organisms, is by sort of co-opting and really taking over the machinery of the cell to make more virus. And so this is done typically by having the viral proteins latch on to cellular proteins and redirecting their activities. And so Nevin and his colleagues were able to do this and to identify a large number of cellular proteins. And it turns out, quite interestingly, that many of these cellular proteins are proteins that we know at the Buck are involved in the aging process. And so this led to the identification of a number, actually 69 potential drugs that we know interfere with these processes that are currently being tested at the Pasteur Institute and at Mount Sinai in New York. And I just got off a call this morning with Nevin, who mentioned that they got their first batch of results, and some of the drugs actually make the virus grow faster, and some others make it grow slower. So hopefully out of this panoply of new drugs and new interactors, we're going to learn more precisely how the virus works and identify sort of its Achilles heel in places that might allow us to suppress its ability to replicate.
And some of those promising drugs involve compounds that have been shown to be involved in the aging process, correct?
Absolutely, and this is quite exciting. This is where I think the history of the buck in conducting research on aging is really incredibly important to this effort. Some of your listeners might be familiar with rapamycin. This is a drug that is currently being explored as a drug against the aging process. It targets a cellular protein called TOR. It delays aging, and this was identified as one of the promising potential drugs. Now, there's some worries about rapamycin because it is also at high dose as an immunosuppressant. But there's early evidence that at low doses might actually have a very beneficial effect on the immune system. Another drug that some of your viewers might know about is metformin or glucophage. This is an anti-diabetic drug that is emerging as having quite powerful anti-aging effects. That also emerged in the analysis from Nevin. So we are busy at work trying to really understand how do these proteins and these pathways and these drugs interfere with the virus processes, and how can we exploit this as an indication for novel therapies. I should say also one word of caution is hydroxychloroquine and actually chloroquine emerged out of that screen as well, and have been shown to be working on the virus in an in vitro system, in a laboratory system. But there's a long way going from these early basic research discoveries to going to actually doing this in a patient and proving that it is not only effective, but also safe. And so I certainly do not want to paint a picture that we are just about to discover the drugs for COVID. I think it's going to take quite a while, but we have no other choice than to go down this route.
So just to cut folks short who might be making a note about metformin or rapamycin, you're not recommending that anybody goes out and tries to get these drugs and to start taking them as a preventive for COVID-19.
Absolutely not. On the other hand, if you are on these drugs, as continue taking them, but I would certainly not recommend. These drugs are exerting their effect in the long term. There's no known beneficial effect on COVID-19, but there are interesting links that make us think that the science that the buck has been focusing on is highly relevant to this virus and to this epidemic.
Before I get into immune boosters, which I know people are going to be interested in, why are males? They appear to be at extra risk for COVID-19. Why is that? Do they know?
The short answer is we do not know. There are a number of hypotheses. This risk for complication, again, it's not the infection. Males and females appear to be infected at the same rates. It turns out that males appear to be more at risk for complications. Now, there was a lot of discussion initially. A hypothesis was that this might be related to smoking. We know smokers have an altered immune function in the lungs. And we know that in many parts of the world, males are tended to be more smokers. However, that data has been questioned. There are other hypotheses that are linked to generally to the male behavior in response to epidemics. They tend to listen less to public health recommendation. Males tend to wash their hands less frequently. They tend to, there's a whole series of arguments that have been made that might explain why males might actually be getting larger inoculum of the virus. There seems to be a risk, an increased risk of complication if you are exposed to large amounts of virus. And we see this best exemplified in healthcare workers tend to be getting sick at a higher rate, at a more significant rate than other people.
So why does COVID-19 seem to affect the lungs so much?
That's a very good question. One of the short answer again is we don't fully know. The virus is an aerosolized virus, so it is transmitted via aerosols. That means its portal of entry is by breathing. So that might be as simple as this. In addition to this, we know that the receptor for the virus, at least one of the receptors that has been identified, ACE2, is actually highly expressed on cells in the lung. So it might be a combination of the two factors. But obviously, we know the virus is not restricted to the lung. There are symptoms of infection in other organs. We know that the lung is the primary portal of entry.
Got it. So let's talk about immunity. Everybody is desperate. Many people are desperate to boost their own immunity to give themselves the best shot against this virus in case it comes in contact with them. What can people do right now to give themselves the best shot at either not getting it, or if they get it, that they're going to come out of it okay?
Well, there are everyone is hoping for a magic bullet. And so right now, it is actually amazing to see the proliferation of advice on supplements and a number of modifications that people are recommending. I think it's important to go back to the basics. And we know from our work and the work of many that the good elements of a good immune response is actually general health. And so the first thing that I would encourage the audience is to pay attention to is what we know actually will create health, and that is adequate sleep. Lack of sleep actually has profound effect on the immune system. Even one bad night can affect your immune system. So adequate sleep, good nutrition, rich in all of the nutritional elements such as vegetables and fruits. So sleep, nutrition, moderate physical activity, which actually might be challenging for many of our listeners. However, in many parts of the country, one is still able to go for a walk. And I've argued that try to make this part of your routine. There are profound effects on physical activity and exercise. And last, so those would be my major areas: nutrition, good sleep, and regular physical activity. These are things that we know make you healthy anyway, but they will make your immune system actually more resistant. The last one that I think we should probably pay particular attention these days is even though many of us comfortable at home, these are highly stressful times, and they're stressful for a significant fraction of the population who have lost their jobs, who are staying at home with screaming kids. So there's a lot on people's mind to be confined in the space, along with the uncertainties that we're all living through. When is this going to be finished? Most of us have at least a relative or a friend who has been infected, or we've watched Chris Cuomo on CNN. So all of this exerts a toll on our psyche and generates a significant level of stress, even if you're not conscious of it. And so this means that stress alleviation techniques that we know work and have also a profound effect on the immune system, I think should be thought about. And I don't have a precise prescription, but meditation, yoga, exercise, calm time, everything that you can do that you have the feeling will actually alleviate the stress level that you are under should be an important aspect of staying as healthy and as strong to resist the virus.
So we got some questions about vitamin D and melatonin. Easily accessible. There are articles out there that sort of recommend upping these two items. What's your take on those, on melatonin and vitamin D?
There's also vitamin C.
Right.
So those three are, it's a difficult one, difficult question to answer. I'll speak a little more about vitamin C because I think that's one that has created more noise. The vitamin C story started with Linus Pauling, who was a Nobel Prize winner, who became convinced in his later days that vitamin C had amazing abilities as an antioxidant, and actually used to take enormous amounts of vitamin C, and also wrote books claiming that vitamin C would prevent the common cold and similar problems. Because of his visibility and his notoriety, there was a lot of work actually on vitamin C. Large number of clinical trials have been conducted. And the conclusion at the end was that vitamin C does indeed confer some limited amount of protection against the common cold and against the flu. It was thought that about 8% less duration of symptoms. So if you have normally a cold that lasts 10 days, it would last 9 days. No effect on mortality, no effect on complications. Now, because of this, there are urban myths and rumors that vitamin C is a miracle antiviral. I think it is clearly not. Nonetheless, clinical trials are underway, especially in China, with high doses of intravenous vitamin C. And as for any other modification that involves a significant dose, I think we need to wait for the results. I would say that most virologists who have dealt with viruses cannot see any reason why vitamin C will have a profound effect. So, but the clinical trials have to be done. It could become something significant. Now, the same arguments can be made about melatonin, vitamin D, and other quercetin and other supplements. At moderate doses, they will not hurt you. Another one is zinc, for example. Taking a small amount of these supplements is unlikely to hurt you. If you're considering taking them at high doses, I would urge caution, because at high doses, some of these supplements can have sometimes untoward effects. And so you want to make sure that you're helping yourself, not actually making it worse. And for those of these supplements that emerge as potentially promising, I can guarantee you that clinical trials will be conducted. And personally, I would resist taking them at high doses until the evidence is there, because we know the potential for harm is truly there as well.
So, because it seems like if you take immune boosters, if your body tends towards a cytokine storm, you want a balanced immune system, right? Not one that's like hyper.
That's an important point. Another area, for example, in which we are working, the NAD boosters. Some of you might be familiar with NR and NMN. These are boosters that increase your level of NAD. Well, at this point, there clearly is an interaction between the virus and the NAD pathways. What we don't know is in which direction. Is it better to have more NAD or less NAD? And so, again, we don't know. The work has to be done. If you're taking a small dose of NAD boosters at this point, I would say there's no reason to stop it, but I would not argue to increase the dose either. So we know, for example, in the aging field, that many of the interactions that we see between drugs and effects are not linear. It's not because you're taking more of something that you're actually going to get a bigger effect. Sometimes there's an exact a sweet spot where you have to be to see the beneficial effect.
So there's been some press recently about an antiviral drug called, I hope I'm pronouncing it right, remdesivir. So what about that drug as a potential treatment for those who already have COVID-19?
So remdesivir is a true drug that is going through the FDA approval process, which is the way that all of our drugs are actually demonstrated to be effective, which is not the case for supplements. So I think we have to make the clear distinction between these true drugs versus the supplements, which live in an unregulated world. Remdesivir was generated actually to fight another virus, Ebola. You might recall a few years back, there was a huge epidemic of Ebola in Africa, and the drug was generated for this. It turned out to be not effective for Ebola, which has similarities. It's not identical. Of course, it's a completely different form of family, but it's of viruses, but it has some similarities. It turns out that remdesivir actually works on a key coronavirus protein, the polymerase, which is the enzyme that the virus uses to replicate its own RNA. And so very strong effect in the laboratory setting, which has led to the idea of testing it in clinical trial. And this is where it is now. We don't have any results, but I think these results should come in the near future.
Got it. So a couple questions about antibody testing. Do you see promise in it? What's the most recent science discovery about the degree and time of antibody? And then there's another question. How realistic is this antibody testing given the fairly low total infection rate and long time frame for trials? So talk about what is antibody testing, first of all, and then what do you think of it?
So I'll step back and actually tell you a little bit of when we talk about testing right now for the virus, what are we testing for? So if you go now for one of the detection centers, and because you have symptoms, you are being tested for the presence of the virus itself. And so we can now, using a technique called PCR, identify the presence of the RNA from the virus. So this is what the current detection tests are. Now, obviously, this only looks at people who are actively infected, productively infected. What we are very interested in having as a tool, especially if we consider moving to the next stage of the epidemic, which is to lift the lockdown and try to go back to a more normal way of living, is to identify the subset of us that has been infected already. And so the way we can do this is by looking for these antibodies. The antibodies, remember I told you about this educated immune response, the part of the immune response that is specific for the virus. So when we get infected by a virus, we generate these antibodies. They have the ability to latch on to the virus and to neutralize it. These are so-called neutralizing antibody. And we can detect them in the blood of people who have been infected. It takes 3 to 4 weeks to detect them. Typically, it is also, for most viruses, not all of them, is indicative of immunity. Immunity means that you are actually protected from a super infection. So the reason we all want to identify who has been infected is you can imagine that these people will be the first one to be able to go back to work by being protected. This would also give us an idea of how close are we to herd immunity. Obviously, we know, depending on different viruses, it takes a different fraction of the population to have immunity to actually confer herd immunity to the rest of the population. And so those numbers vary between 50 and 70%, depending on, for most viruses. Obviously, we're not there now, although I should say that I read a study yesterday from New York, in which they tested every pregnant woman at the New York Presbyterian Hospital. Every woman that came in to deliver a baby in the last few weeks was tested for the presence of coronavirus, and the presence of antibodies. They found that two things: 15% of these women had antibodies, which means we have quite a lot more penetration of the virus, at least in New York, than we had suspected. And perhaps more remarkably, out of these 15% of women, 88% were asymptomatic and had no recollection of having been infected. So this really, again, indicates that the virus might actually be much more distributed in the population that we had assumed. It's both bad news and good news. It's good news because we know that the virus only affects a very small fraction of the population in a really bad manner, the good news is that the bad news is there are probably a lot more infected people than we had suspected. That means we will need to identify all of the asymptomatic shedders, carriers, to be able to go back to a normal situation.
So there's some talk about people who are super spreaders. Who are they?
Yeah, so we've seen this with most viruses. This is not something that's unique to corona, to this particular coronavirus. The word just says it. There are a number of people who are just generating a larger amount of virus and spreading it in a more efficient manner. And the key question is, why is this? And can we don't know. A second key question is, how do we identify them? Because obviously they are the ones who are really contributing to the spreading of the epidemic.
Got it. We got a question online this morning. Somebody wants to know if the Buck is working on a vaccine. I can say no, we're not working on a vaccine. Why don't you talk about what it takes as far as in a lab environment to actually work on and develop vaccines?
So the way vaccines are being made are the classical vaccine. The first vaccines that were generated consisted in taking a virus and making it multiply in the laboratory setting. And when you do this, the virus essentially becomes disabled. In a way, it can still replicate, but it is hobbled. It is. Okay, hold on for 1 second. I realize I have someone calling me. I thought I had shut off my telephone. So let me see, where was I? So how do you make a vaccine? The oldest manner is to disable the virus and to make it replicate in the ways in which it becomes less fit. You can then put it back in people, and eventually this virus does not induce the full disease, but still induces immunity. This is how the first polio virus was generated. Another way is to actually take the virus and inactivate it. You heat it, for example, and then you actually administer it to people. They see it as a virus, even though it's not able to divide, and they still mount an immune response. Now, these are the original way of making a vaccine. Today, we do this typically with specific subunits of the virus, so we can express one viral protein or two that we know are the most important, and actually inject those in people. We can produce them in a laboratory setting and inject into people. And lately, the last way is we can actually inject the viral RNA or the viral DNA directly into people, generating protein. So all of these are currently explored. There are lots of biotech companies and large and small actually pursuing each of these approaches. Hopefully, one of these will pan out and will yield an effective vaccine.
So a question that just came in, beyond today's pandemic, how can we instill preventative self-maintenance of health as a way to decrease the toll of future pandemics? Should we change how we live?
Great question. And I think we should look at what has happened in some Asian countries and what is happening in the US and in Europe. There has been, over the last 40 years, a global disinvestment in a field that we call public health. Public health is the science of understanding how do epidemics happen, how do we monitor for them, and once they start, how do we control them, how do we snuff them out. It turns out that for the last 30 to 40 years, the science of epidemiology and the science of public health, our public health systems, have been not paid attention the way they should be. These represent significant investments, obviously, but they pale in comparison to the amount of money that we have to spend now. So an ounce of prevention in this case is just exactly what we need. For example, think about what will happen when we come out of our lockdown, which will happen progressively in the next few weeks, hopefully next month or two. We are going to need to identify everyone who's been infected. We're going to need to identify everyone who is productively infected. So we will need these tests, but not only the tests. Once we identify positive people, we will need to do something called contact tracing. That means identifying if a person emerges as positive, identify everyone who's been interacting with this person in the last 2 weeks, notify them and isolate them. That's the only way we will be able to control this virus in the absence of a lockdown. And so it's been estimated that we're going to need to hire close to 300,000 people just for the US alone for contact tracing. And obviously we don't need such a workforce on a long-term basis, but if we had a stronger public health, we might have been able to prevent this right from the beginning. And I think hopefully, as a society, we will learn the lesson and move into the future with a reinforced public health system. Because the thing to remember about this pandemic is that there will be more. There are thousands of viruses lurking in animals in different places of the world, waiting to jump into humans and to cause similar or worse problems. So I think we should, I won't say we should count our blessings, but this is not the worst virus that could be doing what it's doing right now. There are other potential pathogens that could be worse even.
So that leads to another question online and one I have. So talk briefly, how did this virus jump from animals to humans? And is it a public health issue to try to stop that from happening in the first place? And I think climate change could be involved in all of it. Why don't you give us your thoughts on that?
With regards to climate change, I think we know that we're in the process of disrupting ecosystems that have been in place for long periods of time. When we disrupt ecosystems, there are untoward consequences that can happen. I am not suggesting that coronavirus is linked to this, but there are many ways in which global warming is thought to lead to such possibilities in the future. Now, how did the virus jump? In this case, it is thought to be from a bat. We knew beforehand that bats contain these coronaviruses and many others. So there's still a potential for others. In this case, it is thought to have occurred during a market sales, but it could happen with a kid going into a bat cave and being bitten by a bat. So I think we will never be completely immune to the fact that viruses can jump from other animal species to humans. I think this has happened. This is how the HIV epidemic started from a chimpanzee. It's a chimpanzee virus that jumped into humans, and it will happen again. So I think trying to suppress all of the jumping event is almost impossible. What we can do is to mitigate what happens when the jump has occurred and to stop it. And this, for example, is what we did in the case of Ebola, in the case of SARS, which was a related virus to this virus, in which we were able to stop it a number of years ago. For some reasons, we were not able to stop this one. I think hopefully we will learn our lesson and be more prepared.
Got it. So a question that just came in Marin County, where we are, as of today, we had 171 diagnosed cases and 121 people recovered. Do the people who recovered have antibodies that might be used for a treatment or in vaccine development?
That's a very good question. So this is another one of the therapeutic modalities that I have not discussed yet. So we have the drugs. I think we're working actively on this. We can hope we will have a drug for the people who are infected. We have the vaccines, but we also have the antibodies. So as I mentioned earlier, some of these antibodies are neutralizing. That is, if you were to receive them from someone who had survived an infection, you might actually be protected already and immediately without having to wait the 3 weeks to 1 month that it normally takes you to raise antibodies. So that's one possibility. There are efforts in this direction. Another way, but it obviously involves some really big complication because you need to identify all of the infected people. You need to extract their blood. You need to purify the immunoglobulin, which is the antibody. So very complicated and expensive process. Another approach is to identify in those people who have survived an infection and who have mounted these antibodies, is to identify the most potent and specific antibodies. And then we can, using genetic engineering, we can actually produce them in factories. This approach has actually been used against HIV, and there are now a number of really highly neutralizing antibodies that one can administer to patients, and they actually will suppress the infection. So we can hope, I know that this type of research is underway. It is one of the many approaches that are being pursued, and we don't know which one will eventually be successful.
Will there be something to learn? There was a question about folks who appear to be immune to the coronavirus, maybe, have been infected but have no symptoms, asymptomatic. Is there something to be learned from them as well?
Yes, absolutely. Again, in the HIV field where I worked for a number of years, we knew there was the whole gamut. Some people actually were exposed to the virus and could not be infected. We identified the receptors, and some people actually carried a mutation in this receptor. We can expect that some people in the human population have a mutation in the SARS-CoV-2 receptor and will therefore be immune. We also know, in general, younger people tend to have less strong manifestations. Why that is, we don't fully understand. This is something that we intend to study. I think we can expect the whole gamut. And this is what we see with many viruses. One important aspect is the size of the inoculum in virology is known to be one of the key determinants of pathogenesis of disease. That is, for some viruses, a small inoculum might actually not yield a very significant disease, whereas a larger inoculum will yield a much more significant disease. We have preliminary evidence that this might be the case in COVID-19.
Got it. So I'm assuming that there are folks that are watching this webinar who are dealing with chronic conditions and are on medications. Maybe some of them are anti-inflammatory conditions like rheumatoid arthritis. What should people do when they look at the pills they take every day and they wonder, gosh, what should I do? Are these good for me now? Or are they bad for me now?
I think they should continue taking their medicine. They should closely consult with their physicians. This subset of people who have chronic conditions, I would argue, are the last ones who should experiment with drugs that are currently rumored to work. We heard the case of a person who actually took a larger dose of hydroxychloroquine and actually died from an arrhythmia. So if you have already existing conditions, you are at increased risk for the complication of this virus, but you might also be at increased risk for the complication of the drugs that you will take. So I would argue that everyone should really closely consult with their physician. And until we have firm evidence, this is not a time for self-experimentation or taking chances on a non-proven drug. We know that drugs can be miracle, and many drugs that have been developed actually have incredibly positive effect. But there are no drugs that comes without any side effect, none. And so when you decide to take a drug, I think you always have to ponder, am I willing to live with the potential side effects? And in the case of hydroxychloroquine, for example, or azithromycin, two drugs that have received the largest amount of publicity with their potential effect on the virus, these drugs carry significant potential complication in terms of heart rhythm troubles. And so I would urge caution. It's not the time to experiment on anything.
So we're getting close to wrapping up, but I want to talk a little bit about the weather. We got a question. Gorgeous sunny day here in Northern California. Temperatures are going to get up into the 70s. Does hot weather make COVID-19 more containable, or is that a myth?
It's at this point, it's a myth. where is the myth coming from? It turns out that a number of viruses exhibit what we call seasonality, and the flu is the best example. The flu tends to occur more during the winter month. No one knows why, by the way. A number of hypotheses have been advanced. Maybe people are more together in enclosed spaces during the winter. That's clearly the case for the flu. In the summer, it's quite rare to see flu cases. There is currently, unfortunately, no evidence of any seasonality in COVID-19. We should have seen it already, although there's some areas where the virus appears to be relatively low, and some of these areas are more in the southern part of the country. There are also some exceptions, like Louisiana. Right now, Louisiana, which has hot weather, is actually suffering one of the worst epidemics. So I would say so far, the consensus is that the seasonality is not evident.
So final question for you. I know it's hard for me to watch what's happening in New York in hospitals, and it's like, oh my gosh, but there is heartening activity that's happening. Can you talk about the science? Like what's happening with the collaborations and the science that is, I think, is a good thing. Can you talk about that?
Yes, and I think when you have Nevin Krogan on next week or a couple of weeks, I think it would be great also to follow this up with him. I think there is science is, for those of you who are not in science, you might not know this, but it is an intensely competitive affair. We are humans, we are passionate by our ideas, and typically scientists sometimes collaborate, but also compete fiercely. What I have seen in the case of the COVID-19 and this coronavirus is really the opposite. And the collaboration that we have with Nevin Krogan and his group and Melanie Ott has just been the most open collaboration that I have ever seen. People bring so there are a large number of groups. I don't know the exact number, Nevin will say, but hundreds around UCSF, the BAC and others. People come to meetings, they share their very preliminary data, they share their reagents. So I think everyone recognizes this is not a time for fierce competition. This is a time for collaboration and bringing a solution as fast as we can. And in some way, it is a new way to do science. I found it incredibly refreshing. And I think it will change the way we work in the future. And hopefully this will be one of the good things that will come out of this epidemic.
That's great. Well, thank you so much, Eric, for being with us today. And I want to thank everybody who joined us. We got some great questions. I know we didn't get to all of them. We will keep them in-house, and maybe you'll see a blog post in the future that addresses some of those issues. A video of today's event will be posted online. We'll let you know when it's up and available. We had hoped that our next conversation would happen this coming Friday, but we just got word this morning that we have to reschedule our session with Dr. Larry Brilliant. The cancellation is for the greater good. Dr. Brilliant is a world-famous epidemiologist who has firsthand experience with pandemics. Working as a United Nations medical officer, he played a key role in the World Health Organization's successful smallpox eradication program. So he's been called away to an important meeting, but we will get that rescheduled. We'll let you know. So I'm going to be back here next Wednesday, April 22nd at 10 a.m. Pacific, 1 p.m. Eastern time. We're going to be talking with Dr. Nevin Krogan from UCSF. As Eric mentioned, he is leading an international collaboration of researchers who are looking at the virus and how the virus impacts ourselves and how our bodies react to the virus. I promise you it's going to be a great session, and I really hope you'll be able to join us. In the meantime, please mindfully enjoy the rest of your day and stay safe and healthy. Thank you so much for being with us.
Thank you, Chris.